Hello, my name is Dara Tolchin, and I will be presenting my poster, De novo SOX6 variants cause a neurodevelopmental syndrome associated with ADHD, craniosynostosis, and osteochondromas. This research was conducted at the Children's Hospital of Philadelphia in the Department of Surgery in the Division of Orthopedic Surgery. The SOX gene family consists of 20 transcription factors that play a pivotal role in cell fate and differentiation during development. These genes contain a highly conserved high-mobility HMG domain that has been shown to be critical for DNA binding and bending, nuclear trafficking, and protein-protein interactions. Mutations within this transcription factor family have been associated with rare congenital disorders known as SOXopathies. These mutations are commonly de novo heterozygous and inactivating and exhibit gene haploinsufficiency. SOX6 is known to be involved in chondrocyte differentiation and the development of the central nervous system. There have been reports of SOX6 variants causing adult pathological conditions. However, there is yet to be a well-established association between SOX6 variants and a developmental syndrome. Therefore, the objective of this study was to use clinical and genetic data to examine SOX6 variants found in 19 individuals that demonstrated developmental delay and to test the transcriptional activity of 4 missense variants in vitro to determine if SOX6 haploinsufficiency leads to a developmental SOXopathy. Expression plasmids for the 4 missense variants were generated by PCR mutagenesis, and they were transfected into HEC293 cells in triplicate and were collected using cytoplasmic and nuclear extracts and prepared for Western blot analysis. Whole cell extracts were also prepared in order to test the DNA protein interactions via electrophoretic mobility shift assay, or EMSA. In Figure 1, clinical findings show that patients exhibited intellectual disability, abnormal behavior, such as ADHD and autism spectrum disorders. In Panel A, you can see that there were dysmorphic features and skeletal anomalies, including mild facial dysmorphism. In B and C, moreover, there were multiple diffuse osteochondromas found in 3 of the 19 patients. Figure 2 shows a schematic of the SOX6 protein. The purple denotes the 4 nonsense variants that elicited zero functional activity, while the blue showed 2 frame shift mutations, and the 4 of interest are going to be the red missense variants, and the 2 within the HMG domain being methionine 605 threonine and tryptophan 639 arginine. The location of these missense variants is highly conserved within the SOX gene family in whole, or at least within the SOXD family consisting of SOX5, SOX6, and SOX13. In Figure 3, functional tests of the SOX6 missense variants showed that all variants were effectively expressed after transfection in HEC cells. However, the methionine 605 threonine and tryptophan 639 arginine proteins were not translocated or retained into the nucleus as efficiently as SOX6, as seen here and seen at this ratio calculated of the nuclear cytoplasmic ratio. In addition, these 2 variants failed to bind the DNA probe in the EMSA and to transactivate the reporter in the luciferase assays. On the contrary, tryptophan 161 cysteine and serine 746 leucine showed normal protein activity. Finally, Panel D shows that none of the variants interfered with wild-type SOX6 activity, suggesting a lack of dominant negative effect of the variants. Lastly, in Figure 4, analysis of SOX6 expression in the human brain. SOX6 was shown to be expressed in fetuses, children, and adults, mainly in prospective brain structures. RNA expression was highest in the ganglionic eminence, amygdaloid complex, and hippocampus, which all play a pivotal role in brain development. In conclusion, the findings from this study concur that SOX6 haploinsufficiency leads to a specific form of neurodevelopmental SOXopathy characterized by mild to severe intellectual disability and in constantly associated skeletal anomalies such as mild facial dysmorphism, craniosynostosis, and osteochondromas. No clear genotype-phenotype correlations were found. This new syndrome has been designated in online Mendelian Inheritance in Man (OMIM) database as number 618971 Tolchin-Lekenyiek syndrome, or TOLCAS. For the complete list of authors on this project, please visit the webpage at the bottom of the poster. Thank you.